Hello, welcome. So today we're going to we're going to discuss about glycolysis. Glycolysis is a very interesting pathway because it is a universal pathway. It occurs in all the human cells, even in most bacterial cells. Basically, all cells do glycolysis, and it's the main common pathway that cells use to tap energy or derive the energy that is stored in the bonds of glucose. So let's see. Welcome. So as I've mentioned, glucose is the preferred source of energy for all cells. And where do cells get this glucose from? It can come from dietary intake. As humans, we predominantly eat starch and glycogen and other forms of carbohydrates such as sucrose in the sugar, sucrose, galactose that you can get from lactose in milk. Yeah, so dietary is a big source of glucose. Of course, this is digested, absorbed, and assimilated into the cells. Another pathway is glycogenolysis. We have mentioned glycogen is stored in human cells, predominantly in the liver and the muscle and skeletal muscle. So breakdown of this glycogen, we refer to it as glycogenolysis, serves as a source of glucose for the cells. Of course, during periods of starvation where the dietary intake is low. A third source is gluconeogenesis. This is the process which cells can use to synthesize glucose from non-carbohydrate precursors. And this does not occur in all cells. It occurs in the liver, the small intestines, and kidney. The liver synthesizes this glucose to supply it to other tissues in the blood. So it releases this glucose. It has synthesized through gluconeogenesis into the cells to be utilized by cells like the red blood cells and the brain. We understand that glucose is not able to enter the cell. The phospholipid bilayer, because it is a large molecule and also it is charged at physiological pH. And so it enters through the cells through glucose transporters. And once it enters into the cell, it can be used through glycolysis for providing energy. And depending on whether there's oxygen or not in the anaerobic or aerobic pathways, its fate, we'll see how it happens to continue providing energy. So it can provide energy both in an aerobic environment and an aerobic environment. So this diagram just summarizes some of the things that happens to glucose when it enters the cell. We're mentioning for this lecture, we're going to look at how it is used in glycolysis. However, glucose can be used in other pathways like the hexose monophosphate chat or the PP pathway. It can be used in the uronic pathway to synthesize glucuronic acid, which is used for detoxification reactions. We're going to see this pathway later on. It can also be used in synthetic pathways in the synthesis of other molecules, like it can be used in the synthesis of other carbohydrates such as galactose and fructose. We're going to see some of these reactions. It can be used to synthesize glycogen in the skeletal muscle and liver. Excess glucose can be used to synthesize fat through the process of lipogenesis, and glucose can provide a carbon skeleton, or metabolites of glucose can provide carbon skeletons for the synthesis of the non-essential amino acids. So even though we are looking at glycolysis, it's just one of the fates of glucose once it enters our cell. There are other functions that can occur depending on the energy need of the cell and also depending on the requirements of the cell at that particular time. Let's slightly look at the digestion of carbohydrates. And we've said when we talk about digestion, we're mainly thinking about starch, but also we are thinking about the disaccharides that we can consume. So the carbohydrate digestion will start in the mouth, where we have the enzyme salivary amylase. Salivary amylase predominantly breaks down the alpha 1 to 4 glycosidic bond that are present in starch. Remember, starch had both alpha 1 to 4 and alpha 1 to 6 glycosidic bonds. However, salivary amylase can only break alpha 1 to 4 glycosidic bonds, and depending on the time it spends in breaking down of starch, it will break the starch molecules into maltose and dextrins. Salivary amylase works in a predominantly alkaline environment, which is provided by the saliva and requires chloride ions for its activity. Chloride ions serve as a cofactor. Because of this, once the food lands in the stomach, which is highly acidic, this acidity inactivates the salivary amylase enzyme, and so no carbohydrate digestion occurs in the stomach. Carbohydrate digestion will resume in the small intestines, where we have enzymes that have been synthesized in the pancreas and released in the duodenum. And one of these enzymes is the pancreatic amylase. The pancreatic amylase continues breakdown of the alpha-1 to 4 glycosidic bonds. However, we'll have a second enzyme referred to as dextrinase, which has the ability to break the alpha-1 to 6 branching points. And the result of pancreatic amylase and dextrinase enzymes is going to be maltose and smaller oligosaccharides or disaccharides, mainly disaccharides. The third level of digestion will occur at the brush border. This is the final level, the brush border of the enterocytes, where we have enzymes we refer generally as disaccharide. These include lactase, sucrase, maltase, and these are able to break these disaccharides, lactose, sucrose, and maltose, into their respective monosaccharides. And so, the combination of the enzyme salivary amylase, pancreatic amylase, dextrinase, and the brush border enzyme gives our end products of carbohydrate digestion in the GIT of glucose, galactose, and fructose. So these are the products that are going to be absorbed. Now, before we talk about absorption, let's look at glucose transporters. We mentioned glucose is not able to bypass the phospholipid bilayer, which makes the cell membrane of all the cells. And so for it to, the factor why it cannot traverse fast, it is water soluble, so it cannot pass the hydrophobic bilayer. 2, it is a huge molecule. It's actually made of 6 carbons, so it's pretty huge, but also it's charged. So all these factors make it unable to traverse the cell membrane. So it has to be carried by transporters. And so if you think about the glucose transporters, we can classify them into 2, whether they are sodium-dependent transporters or the sodium-independent glucose transporters. So the sodium-dependent are referred to as GLTs, are abbreviated as SGLT, while the sodium-independent are referred to as GLUTs. Looking slightly at SGLT, they are classified into 2, SGLT1 and SGLT2. SGLT1 is located in both the small intestine and kidney, and it co-transports both glucose and sodium in both the kidneys and the gut. SGLT2, on the other hand, is primarily found in the kidneys, and it plays a major role in the reabsorption of glucose after the filtration of filtration of blood by the glomeruli. So it plays that important role of reabsorption of the glucose from the renal filtrate into the bloodstream, and this is the one that plays an important role in preventing glucose from appearing in urine. So these two, what they do is we have mentioned it's a co-transport. They can transport both glucose and sodium. On the other hand, we have insulin-independent glucose transporters. And so this one do not do a co-transport of sodium and glucose. They carry glucose along its concentration gradient from a region of high concentration to low concentration. We can classify them into 2, the insulin-sensitive transporters and the insulin-insensitive transporters. The insulin-sensitive transporters, for example, GLUT2, are found in the skeletal muscle, adipose tissue, and heart. And these ones, they transport glucose across the cell membrane in response to insulin. So we can see the skeletal muscle and adipose tissue are not able to take in glucose unless insulin is there to facilitate the function of the GLUT2. Then we have the insulin-insensitive transporters. So these ones are going to transport glucose across its concentration gradient, even in the absence of insulin. They include GLUT1, which we find in the red blood cells and the blood-brain barrier. And this is very important because we know the red blood cells are not able to metabolize any other fuel apart from glucose. And of course, the brain predominantly prefers glucose as a source of energy, although in high extremes like starvation, it can burn ketone bodies. Then we have GLUT2. GLUT2 is present in the liver, pancreatic B cells, the kidneys, and the small intestines. And the interesting thing about GLUT2 is that it can transport glucose either way. Most of these transporters, like for example GLUT and GLUT4, they carry glucose from the blood into the cells, and they're not able to carry it back. However, GLUT2 can carry the glucose bidirectionally, so it can, for example, in the liver, when the glucose levels is high, it can take glucose into the liver, and when the glucose level is low, the liver cells can release glucose back into the bloodstream. Then we have GLUT3, which are predominantly found in the neurons and the placenta, and GLUT5, which is predominantly found in the small intestines and play a major role in fructose absorption. So once we understand about the glucose transporters, we can now discuss the absorption of the monosaccharides following digestion. So if we look at the brush border end, if we think about our enterocytes, glucose and galactose will be carried by SGLT1 through a car transport from a region where they're in high concentration in the lumen of the GIT to the enterocyte, which has a low concentration. So here we have a co-transport of sodium, either with glucose or with galactose. Please note they're not carried all at the same time. This transporter carries both glucose and galactose. So it can co-transport glucose and sodium or galactose and sodium. And as you said, it's through diffusion from a region of high concentration to a region of low concentration. Similar, on the other hand, fructose is carried through GLUT5 from its region of high concentration to a low concentration. So again, this is facilitated diffusion and it is not a co-transport. GLUT5 predominantly transport fructose. Once into the enterocyte, we have to maintain a low concentration of these monosaccharides in the enterocyte to facilitate further absorption. We have GLUT2, which readily carries the monosaccharides from the enterocytes into the capillaries or into the bloodstream. Okay, so maintaining a low concentration. However, remember here there's a co-transport of sodium and the monosaccharide, either glucose or galactose. So if sodium accumulates here within the enterocyte, the concentration of sodium will inhibit the diffusion of glucose, or facilitate diffusion of glucose across into the enterocyte. So what happens is that sodium is pumped out through the sodium-potassium pump on the basilateral membrane, somewhere like here. So you find the sodium is pumped out in exchange for potassium. And because the concentration of sodium is higher outside the enterocyte, it is pumped against its concentration gradient, and this requires energy from ATP. So this is how we maintain a low concentration of sodium to facilitate the co-transport of both glucose and sodium by the SGLT1. Once the monosaccharide are absorbed, they get into the bloodstream. They are carried by the portal vein, which takes all the blood from the GIT, from the guts, from the small intestines, and the first point they reach is the liver. The liver, of course, we know it has the immune function to do surveillance on the blood to check if there are any microorganisms that have entered. But also, besides its surveillance function on the immunity, the liver also plays a very big role in metabolism of carbohydrates, in that it takes up as much as 80% of the glucose from the portal vein. So this is very interesting. So most of this glucose is stored as glycogen by the liver, which can be released later on when dietary sources are low. So the remaining 20% glucose now enters into the general circulation, where it is circulated in the blood and it reaches the different tissues. And so, depending on the kind of glucose transporters, the glucose will enter the cell either in an insulin-dependent manner or an insulin-independent manner. So we do know tissues like muscle and adipose tissue really depend on insulin to facilitate entrance of glucose through their transporters, the GLUT4s. Okay, so our monosaccharides, this diagram is just describing what I'm saying there. Our monosaccharides are absorbed, and in the liver, they are converted into glycogen, which can be released during periods of low sugar. Okay, some of the excess carbohydrates we are going to see are stored as fat, which is stored as adipose tissue, predominantly under the skin, but also around internal visceral organs. As we still the time of deficiency, we can think about what would happen if we have deficiency of some of the enzymes of digestion of carbohydrates. Common deficiencies that have been found are hereditary deficiencies of the saccharidases like the lactase, maltase, and sucrase. Okay, so these are the common hereditary deficiencies that have been seen. Not that they are so common, but they have been found. But also deficiency can come about as a result of intestinal diseases, such as malnutrition, or drugs that may injure the mucosa or the small intestines, for example, anti-cancer drugs. So a good example is when we have the severe diarrhea in an individual, you find most of the brush borders are lost, and this causes a temporary enzyme deficiency, and we find those people who had severe diarrhea cannot drink milk or eat significant amounts of dairy products or sucrose or a lot of sugar without causing the diarrhea to be worse. Why? Because we lack the disaccharides, which will break down the lactose or the sucrose. So we can see how the acquired disaccharide can originate. Then we have a common one, disaccharide deficiency, which we refer to as lactose intolerance. And it is more common among adults, males over 40s in East Africa, with the African, Native American, and Mediterranean descent. So you think about lactose intolerance. There are two types. One can be inherited where the child is born with the deficiency of lactose enzyme, or it can be acquired from the environment where it's primary lactase deficiency. This is the one we see more common in adult men in their 40s, usually of African descent, and it is commonly thought could be as a result of introduction of cow milk early on during the weaning period. So this lactose intolerance appears as abdominal pain and bloating, diarrhea and gas, nausea, usually after consuming milk or lactose-containing foods, maybe like cake that have been baked with milk or something like that. So why does these symptoms appear? One of the things is that once these compounds these foods contain lactose are consumed, lactose is not able to be broken down because we are lacking the lactase enzyme, which breaks down lactose to glucose and galactose. That means lactose accumulates in the gut, and when it gets to the colon, it is broken down by the gut bacteria. So the gut bacteria breaks it down. Among its catabolic process of this lactose, it produces gases like hydrogen and methane and other acids. And these gases, they accumulate in the gut, and they're the ones that cause bloating and discomfort. So you have bloating and abdominal pain. But also remember, lactose will attract water into the gut, and this will further contribute to the feeling of bloating and also diarrhea and abdominal pain because of irritation of the peritoneum. So it's a really discomforting disease. It's really a discomforting condition, and so you'll find many of these individuals really try to avoid milk because of these symptoms. They're very uncomfortable. So how can we manage lactose intolerant? Of course, the obvious one, keep away from milk and its products. okay, such as milk products and cheese. Which a person should avoid them, even without telling them, they'll just avoid them. Recently, we have oral lactase enzymes, which have been found to facilitate or aid in the digestion of lactose. A good example is like consumption of yogurt, which has lactase enzyme present there. This can be a problem because we know the predominant source of calcium for us is from milk. As if when an individual is not taking milk, they're likely to become calcium deficient. Another thing is use of lactose-free dairy products. There are other alternatives such as soy milk, which are plant-based and can be used as alternative in lactose intolerance. So our glucose has now entered the cells. What happens to it? So in glycolysis is a pathway that is used by all tissues to break down the glucose. And the main purpose for glycolysis is to provide the cells with energy in the form of ATP. And pyruvate is the end product. So in glycolysis, we'll see glucose, a 6-carbon compound, will be broken out to 2 3-carbon compounds referred to as pyruvate. Okay, and in the next lecture, we're going to talk about what happens to pyruvate, the effects of pyruvate. But for this particular lecture, I want us to focus in this reaction from glucose to pyruvate, which contains 10 reaction. Besides pyruvate, we also have ATP and NADH. Okay, NADH, which is also a product of glycolytic reaction. So we can classify the reactions of glycolysis into two phases, the preparatory phase and the payoff phase. Let's look at the preparatory phase. In the preparatory phase, here energy of ATP is invested to increase the energy level of glucose, and also ATP, not only does it increase energy level, you can see, of glucose, it also converts it into a form that can be split into two molecules. Let's look at the first reaction. The first reaction, ATP, is used to provide a phosphate group which phosphorylate glucose to give glucose 6-phosphate. This is an irreversible reaction catalyzed by an enzyme, hexokinase. The next reaction is an isomerization reaction where glucose 6-phosphate is isomerized to fructose 6-phosphate. The third reaction is another phosphorylation reaction which is irreversible, catalyzed by an enzyme called phosphofructokinase 1. Again, the phosphofructokinase 1, which is abbreviated as PFK1, is able to add a phosphate group from ATP to give us a compound, fructose 1,6-bisphosphate. This means in the first fructose molecule, carbon 1 and carbon 6 have been phosphorylated. Now the fructose 1,6-bisphosphate is now ready for cleavage by an enzyme referred to as aldolase enzyme. So it cleaves the fructose 1,6-bisphosphate into two molecules, glyceraldehyde 3-phosphate and dihydroxyacetone phosphate. So an aldehyde and a ketone. The last reaction of glycolysis is catalyzed by a triose isomerase, which converts this keto back into the glyceraldehyde such that the hydroxyacetone phosphate is converted to glyceraldehyde 3-phosphate, such that when we go into the second phase or the payoff phase, we'll have two molecules of glyceraldehyde 3-phosphate. So of note here is that we have used two molecules of ATPs and we have split the 6-carbon molecule of glucose into two molecules of glyceraldehyde 3-phosphate. What happens in the payoff phase? Remember we said the purpose of glycolysis was to provide the cell with energy. And in the first phase we have used energy, but in the second phase we will see the energy being produced. So in the first reaction, it's a reduction reaction where glyceraldehyde 3-phosphate is reduced to 1,3-bisphosphoglycerate. So this enzyme is called glyceraldehyde 3-phosphate dehydrogenase. And we talk about dehydrogenase, we said it is removal of electrons. So these electrons from glyceraldehyde 3-phosphate are carried by NAD, the oxidized form of NAD, of NAD, it accepts them to form the reduced form NADH plus H. Please note we're showing 2, 2, 2 because we are remembering there are two molecules of glyceraldehyde 3-phosphate. As we reduce this compound, we also phosphorylate it by an inorganic phosphate from the environment to give us a compound called 1,3-bisphosphoglycerate. 1,3-bisphosphoglycerate is a high energy molecule and it will readily transfer one of the phosphate group to ADP to give us ATP. Okay, so this is a kinase reaction, it's a substrate level phosphorylation where ATP is formed. So our 3-phosphoglycerate again undergoes an isomerization reaction where the phosphate group is moved from carbon 3 to carbon 2 by a mutase enzyme. And then water, the 2-phosphoglycerate loses a molecule of water, okay, to give us another high energy compound called phosphoenol pyruvate. Okay, so this enzyme here is called enolase. So the phosphoenol pyruvate now is a high energy compound that will readily donate this phosphate group to ADP to form ATP with the enzyme pyruvate, with the compound pyruvate as our end product. The enzyme here is called pyruvate kinase. This is the third irreversible reaction in glycolysis. We can see all the others are reversible. So if we just think about it, in the first phase, we utilize 2 ATPs, but in the second phase, in the payoff phase, we have formed ATP at reaction 7 and at reaction 10. And there are 2 ATPs, but if you remember the glyceraldehyde 3-phosphate, there were 4 ATPs. Okay, so we can think about it. There are 2 here and 2 here, those are 4. But also remember we have the NADH, which we have mentioned in the presence of oxygen can be used in electron transport chain and oxidative phosphorylation to make more ATP. So we say the net yield of glycolysis is 2 ATPs because we use 2 and we make 4, so the net is 2. So in the payoff phase, the main thing that happens is we produce energy and NADH. So in glycolysis, we have seen 3 major things happen. We break down a carbon skeleton of glucose to pyruvate. So our glucose is a 6-carbon, pyruvate are 2 3-carbon molecules. We have seen this substrate level phosphorylation where ADP is synthesized at 2 steps. And also we have seen we have transferred electrons in the hydride ion form to NAD to form NADH plus H. In summary, 1 molecule of glucose is split into 2 molecules of pyruvate, and in the process we synthesize 2 NAD and 2 ATPs, and of course 2 molecules of water are lost. There are some compounds which we refer to as poisons of glycolysis that inhibit glycolysis from occurring. For example, arsenate inhibits the enzyme 1,3-phosphoglycerate. Okay. Iodoacetate will inhibit the aldolase enzyme. We saw this one is the one that split the 1,3-bisphosphor, which split the compound into 2, and fluoride, which leads to accumulation of phosphoglycerate by inhibiting the enolase enzyme. So these are some of the poisons of glycolysis. How is glycolysis regulated? So remember I kept on emphasizing the irreversible steps. We saw the first irreversible step was the first reaction catalyzed by hexokinase. We saw the second irreversible step was the one where fructose 6-phosphate was converted to fructose 1,6-bisphosphate, catalyzed by phosphofructokinase 1 enzyme. And the last step, which was another substrate level phosphorylation, where we saw phosphoenol pyruvate being converted to pyruvate, catalyzed by pyruvate kinase. So these 3 steps are the major points of regulation of glycolysis. So let's look at the hexokinase. So hexokinase is the first enzyme that phosphorylates glucose once it enters into the cell. In humans, there are 2 forms of enzymes that phosphorylate glucose. We have the hexokinase enzyme and the glucokinase enzyme. The hexokinase enzyme is found in all cells, all the cells in the body, and it phosphorylates glucose to glucose 6-phosphate. Glucokinase, on the other hand, is only found in the liver. Okay, it is only found in the liver, and it also is very specific for glucose, and it phosphorylates glucose to glucose 6-phosphate. We can compare differences in the activities. We can see even the name glucose is there in glucokinase, because glucokinase only converts glucose, phosphorylates only glucose to glucose 6-phosphate. However, hexokinase can also phosphorylate any hexose, including galactose and fructose. Okay, and it is universal. It is present in most tissues. Another site where we see glucokinase is in the pancreatic beta cells, and we are going to talk about this when you talk about insulin. So what is the affinity? When you think about Km of an enzyme, we're talking about the affinity of that enzyme for its substrate. So glucokinase will only phosphorylate glucose when there's a very high concentration of glucose, meaning it doesn't like that glucose, but when it is in high concentration, it will phosphorylate it. Or hexokinase, on the other hand, it requires very low concentration of glucose, as low as 100 micromole. And so even low concentration of glucose will see hexokinase phosphorylating that glucose. However, maximum velocity, the rate at which the enzyme converts the substrate to product, is much higher in glucokinase. So glucokinase enzyme requires high glucose concentration, and it will rapidly convert it to glucose 6-phosphate. On the other hand, hexokinase will require lower glucose concentration, but also it will convert it to a glucose 6-phosphate much slower. How are these enzymes regulated? In the short term, glucokinase, we've seen the response on glucose concentration. So the higher the glucose concentration, the faster the enzyme will work or it will be activated. Hexokinase, on the other hand, is inhibited by glucose 6-phosphate. So if the concentration of end product, if the end product is not moving out, we will see activity of hexokinase will stop. So what happens when a diet has high carbohydrates? In our long term, when we have high carbohydrate diet, insulin will be produced, and one of the effects of the insulin will be stimulating the liver to be synthesize higher amounts of glucokinase. Hexokinase again is produced depending on the amount of carbohydrate in diet. So the higher the carbohydrate, the higher the amounts of hexokinase. So this is the first step of regulation of glycolysis. The next step of regulation is the phosphofructokinase 1 reaction. We saw phosphofructokinase 1 phosphorylate fructose 6-phosphate to fructose 1,6-bis phosphate. Now the phosphofructokinase 1 is the main regulatory step of the glycolysis, and we refer to it as the rate limiting step. When glucose is phosphorylated to glucose 6-phosphate, its fate is not sealed. It can be used in other pathways like glycogen synthesis or the hexose monophosphate shunt. However, once glucose is converted to fructose 1,6-bis phosphate by PFK1, its fate is sealed. It will go into glycolysis. So that's why we refer to it as the main rate limiting step. Now, phosphofructokinase 1 is an interesting enzyme because we refer to it as an allosteric protein, meaning it has multiple subunits, okay? And in addition to the active site, it has other binding sites which we refer to as allosteric sites. And in these allosteric sites, other compounds can bind and change the overall configuration of the enzyme, resulting in either opening up or closing up of the active site. So that's why here this statement says PFK1 is allosterically stimulated by AMP. So AMP signifies low concentration of energy in the cells. And so once we have AMP, it will bind to its allosteric site and it will cause the active site to open, and the PFK will be more active. On the other hand, PFK1 can be inhibited by high energy level. So what does that mean? ATP has its own binding site on phosphofructokinase 1. And when we have high ATP levels, it binds to this site and it will change the overall configuration of the PFK1 and reduce the opening of the active site. And so because the main purpose of glycolysis is to provide energy, when we have high ATP levels, glycolysis will slow down. Another allosteric inhibitor of phosphofructokinase 1 is citrate. Citrate can be brought from Krebs cycle and or TCA cycle, and when we have high concentration of citrate in the cell, it signifies a high energy level in the cell. And because the purpose of glycolysis is to produce ATP, citrate will go and prevent the synthesis of phosphofructokinase when it gets back into the cytoplasm. We have another very potent inhibitor of phosphofructokinase 1, which is actually fructose 2,6-bisphosphate. So if we think, once fructose 6-phosphate is there, it is converted to fructose 1,6-bisphosphate by PFK1. However, when the concentration of fructose 6-phosphate is very high, a second enzyme referred to as PFK2 is activated. And PFK2 now phosphorylates fructose 6-phosphate to fructose 2,6-bisphosphate instead of fructose 1,6-bisphosphate. Fructose 2,6 now bisphosphate will now come and activate PFK1. And we are saying it's the most potent activator of PFK1. It's more potent than ATP and citrate. And remember, PFK being an allosteric enzyme, it must have a binding site for fructose 2,6-bisphosphate, which binds to it and increases its activity. So PFK1, in the presence of fructose 2,6-bisphosphate, will increase the rate of conversion of fructose 6-phosphate to fructose 1,6-bisphosphate. The third site of regulation is the last step, the pyruvate kinase step. And the pyruvate kinase is activated by fructose 1,6-bisphosphate. You can see this is the fourth substrate. So this fructose 1,6-bisphosphate is able to go ahead and activate the pyruvate kinase. So this is an example of a feed-forward reaction, feed-forward regulation, and this has the effect of linking the two kinases, the PFK1 kinase and the pyruvate kinase activity. So if we have high concentration of fructose 1,6-bisphosphate, it activates the last enzyme in the pathway, pyruvate kinase. Then we have the hormonal regulation. So unlike allosteric or moment-to-moment regulation by the allosteric inhibitors, hormonal regulation is much slower and takes time, okay? It takes minutes or even hours, okay? However, the effects of hormonal regulation are more profound and they mainly involve increasing the amount of protein or amount of enzyme that is present. So hormonal regulation can cause as much as 10-fold to 20-fold the enzyme activity, and this occurs, as we've mentioned, it occurs over hours or days. So one good example we've mentioned is when we have high consumer diet that is rich in carbohydrates, okay, or there is administration of insulin. This insulin will stimulate increased production of glucokinase, phosphofructokinase, and pyruvate kinase in the liver. So you can see this one will take a longer time than what you've seen as the allosteric regulation by either positive or negative modulators. Similarly, if you have a diet or the starvation, we have a diet that is low in carbohydrates, we have the hormone glucagon, okay, and the hormone glucagon is going to decrease the synthesis of glucokinase, phosphofructokinase, and pyruvate kinase, okay. So when we have low insulin levels or starvation, low insulin levels like we see in diabetes, these enzymes that are playing a big role in glycolysis their synthesis will be reduced, and consequently their levels with blood will also reduce. So this is what we see. The hormones insulin and glucagon have an important role to play in glycolysis regulation. As we end the lecture on glycolysis, I want us to look at some of the applications or implications of glycolysis. One of them is the Pasteur effect in glycolysis. So remember we said glycolysis can occur both in the presence or in the absence of oxygen. So the rate and the amount of glucose consumed really depends on how much energy, whether there is oxygen or not. So why? We have seen in the absence of oxygen, only 2 ATPs are produced every time a glucose molecule is broken down. So if metabolism is happening in an anaerobic environment, okay, a lot more glucose will be utilized to provide energy for our cell. On the other hand, in the presence of oxygen, the electron transport chain will come into play, and we are going to see the NADH will be taken to the electron transport chain. We're also going to see pyruvate will also enter the electron transport chain, or the TCA cycle and its reducing equivalents will enter the electron transport chain, and a lot more ATP will be utilized. So not as much glucose will be used in the presence of oxygen, or as we see in the absence of oxygen. So that's where we say about 15 times as much glucose must therefore be consumed anaerobically as aerobically to yield the same amount of ATP. So this increased utilization of glucose, amount of glucose in the absence in anaerobic environment or in the absence of oxygen is what we refer to as the Pasteur effect of glycolysis. Another thing I would like to look at is glucose metabolism in cancer cells. We know cancer cells, they grow very fast, okay? And one of the things we have found is that glucose uptake and glycolysis proceeds almost as high as 10 times faster in solid tumors. Okay, so we do know one of the things about cancers is that the cells grow and many times they outgrow the rate at which capillaries or blood vessels grow, such that you'll find big tumors and they're very poorly vascularized or they have very few capillaries. So that means these tumors are growing in the absence of oxygen and they tend to experience hypoxia. Okay, and so the metabolism will be anaerobic. Will use anaerobic metabolism, anaerobic glycolysis for much of their ATP production. And because we're going to see in the next lecture, anaerobic glycolysis will resolve to lactate as an end product. So one of the things we see in cancers is that we have lactic acid being produced. The levels of lactic acid in blood increase. Okay. So another thing that could contribute to increased glycolysis in tumor cells is because of the tumor cells, many of them have smaller numbers of mitochondria, okay? And so there is less use of oxidative phosphorylation and the electron transport chain. Interestingly, they have found that some tumor cells even overproduce glycolytic enzyme, including hexokinase. Okay, and so the rate of glycolysis is much more. Remember, these cells are growing fast, so they require a lot of ATPs, and that means they also require a lot of glucose. If you remember what we just talked about, Pasteur effect. So this is an interesting finding, an interesting biochemical change that we see in cancer cells. As we end, let's look at some of the feeder pathways. We've talked about when we digest carbohydrates, we end up not only with glucose, but also fructose and galactose. What happens to this glucose, or what happens to this fructose and galactose? So somehow these pathways find their way back to glycolysis. For example, if we look at fructose, fructose can be phosphorylated by hexokinase into fructose 6-phosphate, and we say this is the third metabolite of the glycolytic pathway, and so it will continue into glycolysis. Okay, so this is how fructose enters in the glycolytic pathway, mainly in the muscles and the kidney. This differs slightly in the liver, where in the liver we have a specific enzyme called fructokinase that specifically phosphorylates fructose to fructose 1-phosphate. Then the fructose 1-phosphate in the liver will be split to glyceraldehyde and dihydroxyacetone phosphate, which will clearly enter the glycolytic pathway. And the glyceraldehyde will be further phosphorylated to glyceraldehyde 3-phosphate, and this is where they enter the glycolytic pathway. You can see this is slightly different. While in muscle it entered at this point, fructose 6-phosphate, after phosphorylation by hexokinase. In the liver, we have the specific fructokinase. Then what happens to galactose? Galactose is predominantly coming from the milk product from lactose. And in the liver, galactose is phosphorylated by galactokinase into galactose 1-phosphate. Then galactose 1-phosphate is then converted to glucose 1-phosphate, involved in reactions that involve UDP glucose. You can see UDP glucose comes in, it interchanges the glucose for galactose. Okay, so you have glucose 1-phosphate and the UDP is converted. Okay, so we have UDP galactose. Okay, and so the UDP galactose undergoes through some steps, like you can see UDP galactose epimerase, and these several steps eventually convert the glucose to UDP glucose. So defects in this particular pathway has been associated with development of cataracts in infants, to poor growth in children, speech abnormalities, mental deficiencies, and liver damage depending on which enzyme is deficient, from the galactose enzyme to the uridyl transferase enzymes or to the epimerase enzymes. Okay. And mannose can come from digestion of several glycoproteins. We have seen it's present in glycoproteins. And again, mannose can be phosphorylated to mannose 6-phosphate. And we know mannose is an epimer of glucose, so an isomerase enzyme will convert mannose 6-phosphate to fructose 6-phosphate, which will now join the glycolytic pathway. We can see here. Hexokinase can phosphorylate mannose to mannose 6-phosphate, and the isomerase enzyme can convert mannose 6-phosphate to fructose 6-phosphate, which can now continue into glycolysis. So here we have covered glycolysis. We have looked at quite a bit. Started from digestion, we assimilated, we absorbed the glucose, we entered it into the cell, and we have broken it down to pyruvate. In the next lecture, we are going to look at what happens to pyruvate. Thank you for your time.